So this topic you have gone over it maybe twice with respect to your this assignment that I had given you. So I'm sure that you are very much comfortable with the term called fusion tags. So fusion tags are the tags which are set in fusion in frame with your gene of interest, and these can be present either in the N terminal end or in the C terminal end. So, so maybe a little bit recap of what fusion tags are. Maybe your introduction to comparatively more number of fusion tags, rather than the strategy how they are attached, and the various uses of fusion tags is what I'll be telling you about. So fusion tags, fusion proteins in which coding region of your gene of interest is fused in frame to the coding region of the target. Any two proteins or any two entities, it can be a peptide in frame with your gene of interest. So then we say that it's a fusion protein. Which are the various advantages of fusion gene fusions? I'm sure that you have been told of some of these for sure, but others I'll just introduce you to. So the advantage of, because otherwise, if you have to overexpress your gene of interest, then just that would have sufficed. But in many cases, it was realized that addition of a fusion tag either at the N terminal end or at the C terminal end was helping with many scenarios. Which scenarios are those? One is that it was found that if I was adding a fusion tag at the N terminal end, certain fusion tags, not all fusion tags, then they improve the translation of the protein manifold. When I say manifold, it's really manifold, in some cases tenfold. There are many, many examples if you read through the literature, sometime ranging from threefold, fivefold, tenfold, hundredfold, and so on. So that's why the knowledge about various fusion tags and their respective advantages is very, very important. So in one case, it was seen that it is improving the translation. And why we need that is because of the fact that mostly in E. coli, we are overexpressing proteins from eukaryotic origin. So if they are from eukaryotic origin, it was found that for certain proteins, the translation was not very efficient. It could be because of the presence of certain nucleotide sequences which are present towards the N terminal end. So it was realized that if I add a fusion tag at the N terminal end, which is from prokaryotic origin, then somehow it improved the translation. And the addition of that tag, it ensured that the translation will be very good, irrespective of which protein you are cloning. So one major advantage is efficient translation. Then it was also realized that the addition of this tag, it improved the stability of the protein. I'm surprised that one point is not written, and I'll definitely, in fact, I'll add it now. Solubility, solubilize. So I want to say that it was also seen that addition of these tags improved the stability of the protein manifold, and in many cases it was seen that the addition of the tag, it solubilized the protein much more, because whenever we are overexpressing these proteins in bacteria, so it was seen that because we have added very strong promoter and translation is taking place at a very fast pace, so we have a lot of protein being formed. So because these lot of protein is being formed very fast, so there is a chance that the proteins are not folded properly. Because they are not folded properly, they almost then they end up in the insoluble fraction of the cell, or also called inclusion bodies, and then it becomes very difficult for these to be purified further. So in those cases, addition of these fusion tags, they helped in solubilizing also, and also in stabilizing the peptide, means increasing the half-life. So these are two different points. So please remember that. And then it was questioned that how it might be solubilizing. So it was realized that in some fusion tags, it was found that probably they are themselves having some chaperoning activity, while in some cases it was realized that addition of that tag, it was promoting the binding of certain chaperone proteins. In some cases it was seen that the addition of the tag, it was leading to means folding in the form of a micelle, in which the solubility was better. In some cases it was seen that the charge, there was a change in the charge, and that's why the solubility was better, and so on. So there are many detailed reviews which are talking about the various tags and their respective advantages and the respective mechanisms, etc. So one good article is written here. The link is given. Please go through it. It's very good with respect to the gene fusion and the advantages and some of the new fusion tags which are in use and why they are in use, etc. Then these tags are also added because some of these, they can also act as signal peptides. That is, they can direct the protein to a particular place within the cell. And mostly it is the secretion signal, because if your protein is overexpressed and it is secreted out of the protein, then obviously it becomes very easy to purify the protein. You don't have to rely on these breaking the cells and then taking out the protein from the whole cellular lysate, but rather you can just harvest the supernatant or the media in which the cell is growing, and from there you can harvest your protein. So these many secretion signals are also used when you are adding these, when you are doing these recombinant experiments. Then one very important point is the purification becomes extremely easy. And Does that mean that if you have your protein of interest which doesn't have a tag, can it not be purified? Yes, it can be purified. You can use the specific antibody to pull it down. You can use, there are many polyclonal and monoclonal antibodies which are available in the market, which are extremely specific in nature. So from your whole cell lysate, if you want to pull out your protein of interest, you can use that antibody. That antibody will specifically bind to this protein, and that antibody can be bound to some solid support. It can be a bead, it can be a column, and that's how you can purify your protein. But what about those labs in which they have lot of proteins to be purified? One point is this: will you buy a specific antibody for each of the protein that your lab is working on? Or in many cases, there can be a scenario that you want to pull down a protein, purify a protein, but the antibody is not available in the market. Or let's say if it is available in the market, it is not specific in nature. When you are actually working on protein and antibody interactions and working on various antibody, you will realize that for many proteins, we still don't have antibodies in the market. And if they are available, they are not very specific. Okay. So in those scenarios, definitely having a gene, this tag, let's say a histidine tag or GST tag, or so many tags are there. I'll just come to that list. You can use those tags. You can, let's say you're working on 10 different proteins in the lab. You need not buy specific antibodies against it. You can just put a histidine tag on each of those. And if you want to purify it, you can pass it through a nickel column. So histidine nickel thing, I think that might have been already discussed in your lecture on assignments. Then another advantage is protein localization. So if you add some tag which gives a fluorescent signal, then obviously it is very easy to monitor the movement of your protein within the cell. through confocal microscopy or fluorescence microscopy. So what you have to do is just make fusion protein, let's say a GFP fusion with your gene of interest, and then you can monitor it anywhere in the cell. And I have some very good slide for the same, which I'll show you. Then lastly, gene fusions are also very, very widely used for doing the protein-protein interaction assays. So if you want to check, let's say you hypothesize through, let's say, some in silico work that two proteins, they do have very high chance of binding to each other, direct binding. Then how you can test it? You can put one tag in one protein and another tag in another protein, and then do some pull down assays. How you do that? I just that will be my last slide for this today's lecture. So through this slide, I'm sure that you can now understand that what are the various advantages of putting the various fusion tags for a particular protein. So is there any one fusion tag which is the best fusion tag? No. For depending on your need, you have to decide which fusion tag you have to use. If you want to monitor the movement of the protein within the cell, then obviously you will not go for histidine tag. You will go for something YFP, RFP, or GFP, any fluorescent tag that you have to use. If you are facing challenge in the solubility of your gene of interest, then you will preferably go for the tag which has been very widely demonstrated to improve the solubility of your protein. If you want to, having a trouble, you feel that the translation is not very good, then probably you'll go for the tags which are very well demonstrated to improve the translation. And if you want to secrete, means the idea is mainly that your protein expression, etc., all is perfectly fine. It's just that you want it to be secreted out of the cell, then you'll use a secretion signal. And if you want to purify your protein of interest, then you will use certain other tags. But is it that all tags only have one specific advantage? No, there are some tags which can give two different advantages at the same time. Let's say they are also good in improving the translation as well as solubilization. So depending on your need, you will use the various fusion tags. So this is just a very small list of the various fusion tags which are available in the market. And as you can see, these tags, they come with different sizes. Some are very bulky fusion tags. Some are very small fusion tags. Then coming to the function, some have been demonstrated to be very good fusion tags for purification because they have excellent affinity towards particular moiety. For example, histidine for the nickel ions, right? That you have already studied. So these are very good affinity tags. Then it was realized that if you add thioredoxin, I'll just come to one example also, that increases the solubility of your protein of interest many folds. There is SUMO, and then there are so many which are given in that article, that the link for which I've given you. Then I also told you that there are certain tags which have two different properties. So you can add those tags also. Then is it that mostly people go for just a single tag when they are going for, let's say, protein purification? No, in many cases, people also go for double tags. So why double tags? Because it will ensure that your final protein will be extremely pure. So let's say if I put two different tags, sorry, this is some other example, I'll just come to that. Let's say I clone my gene of interest downstream of two different tags. One is strep tag and one is polyhistidine. And I purify my protein first with the help of nickel ions. And then I use that purified protein to further purify it using streptavidin and means this biotin streptavidin activity affinity. Then obviously there are chances that if any unwanted protein was purified because of some, let's say, weak nickel ion binding activity, that will definitely now be removed because now I'm using different affinity tag. So that is, let's say, stepwise purification using two different tags for one protein of interest that you are interested in. So there are many labs and many commercial vectors which are available, which will have two different fusion tags. Okay. Then this example is given that combinatorial fusion tag, in which you add two different tags, one which improves the solubility and one which increases the affinity. And then the article the link for which I've given you, it talks about another fusion tag. I think its name is FH8. And they are saying that it is much better, much better than many of the tags which are already available. And its source is they were working on some parasite protein, which was important for infection. Then they came across this 8 kilodalton FH8 protein, which was important for infection. Then, when they were actually doing the experiment on FH8 uh, production in E. coli for their some, not for commercial purposes, but just for their basic study, they were overexpressing it and then seeing some other effects of it. Then they realized that it was extremely at very high concentration when the protein was being produced. It was extremely soluble. The protein was still soluble, present in the soluble fraction, and also it was very stable in the context that when the cells even at like 74 degree or 75 degree that they mentioned, it was extremely stable in nature. So one advantage was that extremely high solubility and stability, which is provided by FH8 tag as compared to the other tags maybe. And second advantage was the fact that when they were doing further basic studies on that tag, they realized that it has an extremely good affinity towards calcium. So it's a calcium binding peptide. So it means that besides it being used as a tag which improves solubility and stability, it can also be very well used for affinity purposes. I mean, purification purposes. So these advantages are given for that particular tag, and they are saying that because it's very small in size and all the properties that I just told you, it's very good. And why did I tell you this particular example is because of the fact that it's only through the basic studies that you finally come across some component which can be used for commercial purposes. So now FH8 or let's say polyhistidine also must have been some very basic study that was done in the lab. And when people realize that, okay, histidine is a very good affinity tag or whatever, or for sumo or whatever tags are there. So again, it's a dynamic field. It could be that any of you who are working on any particular protein, you may come across a particular moiety which is very good for a particular purpose. Maybe let's say if you want to um, put your protein with it, or let's say express your protein only in the nucleus, and you want to introduce a nuclear localization signal. It means you want to look for a domain which provides the ability for a particular protein to go to the nucleus. So maybe you're working on a protein and you come across a NLS which is very efficient for taking that protein to the nucleus, or you come across a protein, some domain in your protein of interest, which is very good for affinity purposes, and so on. So I just want to say through these examples that be vigilant and try to see that whatever protein you are working on, that what all properties it has and whether they can be used for any commercial purposes or not. So this I've already told you that there are many tags. The examples are given here. You see so many examples are there. People use different secretion signals. And why do they want to use the secretion signal? Is mainly because the protein can be easily purified, and because your protein is now being secreted out of the cell. So obviously it will be little bit this protected from the various proteases which are present in the cellular environment. So one example is also given in which the stability of a particular protein in which a secretion signal was added was increased 10 times. And from commercial point of view, or even let's say if you're working in the lab and you want large amount of protein, this is extremely important. Then actually all these details that how you actually put a signal peptide infusion with your protein of interest has been already dealt in various lectures with respect to the assignment. So this I'll just skip. And another example is of thioredoxin. So if you remember in my previous slide, I had shown that thioredoxin is one moiety, one fusion tag, which drastically increases the stability of the protein. It is very good for that. And also it increases the solubility of the protein. So there are certain examples in which the solubility of a particular protein was increased manyfold. And those examples might be given in the review that I've shared with you. Now coming to that, once you add these tags, how do you purify it? So again, it depends on which tag you are using. There are certain tags which can be purified with the help of metal ions. There will be certain tags which can be purified using other protein binding. Let's say streptavidin and biotin binding is very well known. So if your protein is biotin tagged, then you can purify it using streptavidin in the immobile or in the column or in the bead where streptavidin beads are there, and you pass your whole protein lysate through that. So obviously biotin will bind to streptavidin and then your protein can be purified. In many cases, there are antibodies which are available to the fusion tags, which I'm talking about, which are commercially available and have shown to be extremely specific to these particular tags. So if you have to purify them, you use those antibodies, you attach those antibodies to the inert matrix. It could be the beads or the columns, and then pass through the protein lysate or the supernatant, the secreted protein through that column, and then further purify. So there are, let's say, some calmodulin binding proteins, or let's say I talked about that FH8 fusion tag, which binds to calcium. Then there is maltose binding protein, so it will bind to maltose. So depending on which fusion tag you are using, you will choose that how do you purify your protein through affinity chromatography, which is very, very commonly used for purification. So one example is given, but I think you must have already understood the idea behind it. So here there is this interleukin 2. This is your protein of interest. You made it infusion with this marker peptide. Then you have this column in which you already bind this anti-marker peptide antibody. So then you pass your protein lysate or secreted protein mixture through this particular column. So obviously, because of very high affinity of this marker peptide to this particular antibody, your protein will bind to it, while rest of all the proteins, they will just flow through. So then your protein will bind to it, and then it can be finally eluted. Then comes the question that how do you actually elute your protein? So again, there are areas, I don't know whether I have a proper slide for it, but then you generally use competition between your protein of interest and the another peptide which shows much higher affinity to, let's say, this particular antibody. Or in some cases, you use different pH to elute your protein. So depending on which chemistry you are using for binding for affinity chromatography, you will decide that which method will you use for elution. Then for example, another case is given. In this case, the columns were used, and in this case, the only difference is that the bead is used. So in this case, my protein of interest is showing, it is a fusion protein with glutathione S transferase. It's a GST fusion protein. So these are because it binds to glutathione. So I have these glutathione agarose beads. So my protein of interest will bind to it. And now when I want to elute my protein out of it, then I will add extra glutathione so that my fusion protein gets eluted out, and these uh, uh, it gets eluted out. So, like this, there are so many examples. Please, you can go through these and understand that how these are related. Then comes that I will revisit this lower part, which is regarding the removal of the fusion tags. Or let's say I will just discuss it here only. See, when we add the fusion tag, in many cases, not in all cases, in many cases, you would want the fusion tag to be removed. In which cases you would want it to be removed? For example, if you want to use your protein of interest for therapeutic purposes. So if you want to use it for therapeutic purposes, obviously you will not want any other moiety apart from the protein which is required for therapy, right? So in those cases, you have to remove the tag. So how will you remove the tag? You will remove it by various strategies. One of which is using cyanogen bromide. So what it does is that it cuts at the methionine residue. So in these cases, what you do is that only in those cases where you are very sure that you do not have any methionine in your whole protein sequence, only in those cases you can use this strategy, because it is very simple. You will just treat your purified protein once you have purified it by this treatment with cyanogen bromide. So cleavage will be specifically at this position, and your protein of interest will be all free. But in many cases, you cannot use it because there are chances that methionine can also be present in the body of the protein. So in those cases, what you do is that you add between your fusion tag and your gene of interest a protease cleavage sequence. Okay, so in those cases, you have to introduce in fusion another peptide sequence which can be recognized by certain proteases, which are extremely efficient in recognizing the sequence which is given here, and then making a cleavage. Once they make a cleavage, then this fusion partner is away from your gene of interest, and your gene of interest or the protein purified can be easily used for therapeutic purposes. So, which are the various so this is just showing that what is the sequence. So first we have this 5' leader sequence, then you have this Kozak sequence which has the AUG, and then you have your signal sequence which can be used for secretion, etc. Then you can have your affinity tag, and after that you will have your proteolytic cleavage site, and then you will have your gene of interest. So once you purify your protein, then only you will cut it with the proteases, which will recognize this whole sequence, and your gene of interest will be free. This is a list of only some of the very commonly used tag removal or these protease cleavage sequences which are given. So it means that, let's say you have a flag tag. So after flag tag, you will add this sequence. And after this sequence, in Fusion, you will add your protein of interest. So flag tag will be in Fusion with this sequence, and which will be in in frame and in Fusion with your protein of interest. So you have to take care that the frame has to be maintained starting from your signal sequence or your this affinity tag, whatever you are using. So these are the various recognition sequences that can be used. And these are recognized by certain proteases. So once I've already told you that you treat it with the proteases once your protein has been purified, and then it makes a cleavage and you get your protein of interest. But again, I would like to emphasize that, do we have the best tag removal system? And even if we have best, are we always successful in removing the tag efficiently? Maybe not. In many cases, it was seen that certain tags in certain scenarios were giving incomplete cleavage. In many cases, let's say, even if the cleavage was proper, it was difficult in separating the protease and the tag from the native protein while purifying. Then, in some cases, it was realized that once you treat it with the protease, then there was loss of solubility of the original protein. Again, it was coming, it was not much soluble when you wanted to use it for further purpose. Or while through this whole process, you lost a good amount of protein for final purpose. So these are certain challenges which you will realize either if you're working solely on these sort of projects in your lab as a PhD student, or you're working in an industry and you're given a task of purifying a protein. So at various steps, you have various challenges. So there are people who are working for many years for purification of single protein and further optimizing it and so on. This is just example of another protein cleavage factor that is used, which is this factor XA. So it's factor XA. It is derived from blood clotting factors. That's the history. But for now, I just want to tell you another example. I think now you're more clear already, but still I just tell you, this is tag sequence. Then you have factor XA, which is the site where the cleavage will take place. And in the MCS, you can clone your. So means there are certain vectors which are available in which all this is already there till factor XA. And after that, there is multiple cloning site where you can just clone your protein of interest. You just have to ensure that it is in frame with the tag and the factor XA. So factor 10, sorry. So once you have cloned it here, then because the tag which is used here is a biotin tag, you can pass it through a column chromatography where streptavidin is already bound, and then you get your purified protein. Then you cleave it with factor 10A, and then you get your purified T. So in this whole, many examples are given. So now coming to another advantage of the fusion tag, which was the localization of your protein. Let's say in many cases, you are very curious to know where your protein is localized. So what I will do, I will, let's say I'm interested in one protein, let's say lamin AC. And I will clone it. I will make a GFP fusion of that particular protein. So once I make that GFP fusion and I transfect my cells, let's say in this case it's mammalian cells, I transfect it using lipofectamine, my protein of interest, my vector into these cells. Then what will happen? My vector will take my recombinant protein inside the nucleus. Sorry, my plasmid will go inside the nucleus. There it will We transcribed and translated. Now my translated protein laminase is in fusion with GFP. So wherever my protein is localized, I can see it by fluorescence microscopy or through confocal microscopy for a better look. And I can easily see that the proteins which are nuclear membrane localized, they are showing a very beautiful, localization there. Then there are this histone protein antibody. Sorry, it is shown in fusion with GFP. So you can see it is localized in the nucleus. There are certain proteins which are present in the, that example is not shown here, but there are certain proteins which are present in the cytoplasm. So you can see that it is present in the cytoplasm. You can see if your protein is ER localized through just making these fusions with some fluorescent tag. So in this case, it's a GFP tag. Are there other tags available? Yes, of course. There are RFP tags, there are YFP tags, yellow, red, and so on. Okay. So one very good advantage for using these fusion tags is definitely the localization. And also, in many cases, you would like to see, let's say, in response to a particular stress, that proteins, they move from one place to another. So it could be that in response to treatment with the transcription factor, or let's say treatment with a particular cytokine, or let's say if your cells are exposed to heat shock or some other shock, you want to know whether your protein, it moves from one place to another. So you can see that under microscope. You give that treatment and you will find that, let's say, before treatment they were present in the cytoplasm, but after treatment, let's say exposure to hypoxia or cytokine, your protein now moves to nucleus. So these are very important observations that you can make just by making these fusion tagged proteins. Now, the another example, another advantage is that, can I, let's say, if I want to, I hypothesize that probably my two proteins, they are interacting with each other. So is there some way that I can actually demonstrate it? Yes, I can demonstrate it by co-localization study. So it will at least give me a hint that if I'm saying that my two proteins are interacting, it is but obvious that they have to show co-localization within the cell. Only then they can directly interact with each other. So what I do is that I make one protein as tagged with green, means green fluorescent protein, and another protein that I'm hypothesizing will interact with this with a red fluorescent protein, right? The way you have done with GFP, same way, it is just a different sequence. You will put a tag for RFP. And then under microscope, you do the merging of the same, right? So means you take a picture of the cell in showing a green fluorescence, and take another picture of the same cell without moving it for red fluorescence, and then you merge the two images. If the two images show color localization of the same, you will see that green and red together, they will give a yellow signal. So here I'm very sure that 89% of my cells, which had either green or red fluorescence, they showed co-localization, right? So it means that likely my proteins may be interacting with each other. So again, in many molecular biology experiments, this is a very common experiment that is done, in which through which you use show co-localization. So, oh, sorry, that is the last slide, which is also very, very important, where you want to directly demonstrate protein-protein interaction. Okay, so while this is in vivo, you can show that they are just co-localizing, so likely they are interacting. But here this experiment is done in vitro. So what I do is that, let's say I want to demonstrate that my proteins A and B, they interact with each other. Okay, so I make my A protein fused with flag tag, and B protein, let's say, fused with histidine tag. Okay. Now what I do is that in the cell, I transfect both of these constructs. Okay, एक plasmid में I'm able to express flag tagged A protein, and in another plasmid I have histidine tagged B protein. Both of these plasmids I co-transfect in my cells. Okay, so then what I do is that I once I transfect, after a few hours, maybe 24 hours, 48 hours, whenever you want to time it, you you isolate the whole protein lysate. You just lyse your cells. So what you have a protein lysate. Now I want to pull down my flag tagged A protein with the idea that if my A and B protein are interacting, then by pulling down through the flag tag will also pull down my B protein, right? So what I do is that I use these some this what is this beads which have some tag which can pull down the flat tag. So once I pull down through that, then I run my protein, the purified protein, because now I'm in a way purifying my flag tagged protein through a method of affinity purification. Then what I do is that I do the Western analysis, which detects the presence of a protein using histidine tag. So if my protein is actually interacting with the B protein, then it should give me a histidine tag uh, band with the histidine antibody. So if these two are being pulled, then obviously histidine tagged protein will be in association with it. So that also will be pulled down. And now if I want to see that my pull down protein does have a histidine tag or not, that I can do through Western blotting. And what I will see that, okay, I will see a strong band here. If I see a strong band here, then I can easily conclude that yes, my protein A and B interact. But if I do not see a band here, then I can easily conclude that my protein A and B, they are not interacting with each other. There is no direct interaction. So is this experiment clear to you? If not, then I'll explain again.
Ma'am, I have a question. So here in this case, we are relying on the fact that even after we purify the protein using one antibody, the other
Yeah.
protein, let's say it was affinity purification. So
Yeah.
even when in the collar, the second protein is not dissociating with the first one. So
Hmm.
could this be a source of error when we are actually interested in purifying just protein A?
Yes, it could be. So then you'll have to figure out various conditions in which you can, break the interaction between two proteins. But this is a very, very common technique in those labs which are interested in molecular biology. They want to identify the interacting partners and all. So which techniques will you go for? You will definitely go for immunoprecipitation. Co-immunoprecipitation is the name of the technique, or pull down assay is another name of the protein, in which you are pulling down one protein and that pulls down the other protein. And then so it is all under which condition you are pulling it down. Are you disrupting its interaction with other proteins or not? So uh, in many cases you are very well aware that there are multi-protein complexes which are consisting of 20 proteins, 30 proteins, and so on. So in those labs there are other methods, more gentle methods to pull down the proteins which are interacting with one particular protein, and then it all has to be standardized in that lab, that how you can pull down the whole protein complex and not just one protein. So it is the condition, maybe pH and washing conditions, denaturing conditions, and so on. uh, is it clear?
Yes, ma'am.
Okay, so any other question? Okay, so if there are no other questions, so then we are done. I'm so glad we are finally done with these vectors and purification and proteins. So now we'll start with certain other means methods which are used in molecular biology. So we'll be touching upon sequencing methods, how do you detect transcripts, etc. So other topics in this course. So we'll be moving on to that. But now I think you have got a very, very detailed understanding of how do you overexpress your proteins in various different hosts. So we have touched upon bacteria, yeast, plants, and mammalian cells. So, which are the various vectors that you choose, and which are the various promoters that you choose? Which is the method of the transfer of your vector into the target cell? So, we discussed in mammalian cells which method is used, in plants, which methods like agrobacterium or particle bombardment in bacteria are different, in yeast different. So that means it was a comprehensive introduction to the various methods for protein production in various different hosts. So now I'm done with it. And if there are no more questions, then have a good day.
Thank you, ma'am.
Okay,
Thank you.
guys. Bye bye. Bye. Take care.
Thank you,
Thank
ma'am.
you.